Hi viewers, this is Swamy, Assistant Professor of Zoology. Today we are going to discuss the digestion of lipids. Under this topic, we will discuss introduction to digestion of lipids, composition of lipids, lipase enzymes which digest the lipids, digestion of lipids in mouth, in stomach and as well as the small intestine. First of all, in our food, we will take lipids either in the form of fats of animal source and oils of plant source. The fats from the animal source are in general the butter, cream, lard. Lard means the fat of pig, and tallow. Tallow is the fat of other animals, meat. All these include the fats of animal source. And if you come to the oils from the plant source, all the kinds of plant oils like olive oil, groundnut oil, sunflower oil, sesame oil, and all these oil seeds, various kinds of oil seeds. Chemically, fats are known as triglycerides or triacyl glycerol. Triglycerides or triacyl glycerol. So the meaning itself indicating that one glycerol molecule bound to 3 fatty acid side chains, otherwise 3 fatty acid chains, and due to the fusion of glycerol and the fatty acids, there will be a removal of 3 H2O molecules, water molecules, and finally there will be a formation of ester bond in between the glycerol and the fatty acids. Glycerol and the fatty acids bound together through the formation of an ester bond. Of about 60 to 150 grams of fats has been ingested generally by the human beings, and they will be in the form of phospholipids, cholesterol and the free fatty acids. So they are not at all the polymeric, but they have the short chain of fatty acids. They are in the form of short chain of fatty acids. And the enzymes which are essential for the digestion of the lipids are known as the lipase or lipases. They divide or break down the lipids into glycerol and the fatty acids. Right? Based on various reasons, due to various reasons, the fats, otherwise the lipids, may not be digested completely. 100% of digestion is highly impossible, and thereby the end products of the digestion of lipids are fatty acids, glycerol, and along with them we also find out the monoglycerides and the diglycerides. Previously we have seen all the carbohydrates finally they get converted to monosaccharides, and all the proteins they get converted to amino acids. But here not only the fatty acids, but we will see monoglycerides and the diglycerides due to the incomplete digestion of the lipids. See the structure. Here it is a glycerol molecule, and these 3 are the fatty acid chains, and here H from the glycerol, OH from the fatty acids has been removed in the form of water molecules, 3 water molecules, and here we will see there is a formation of the bonding in between glycerol and the fatty acids through this bond, CO bond. This is nothing but the ester bond. This is nothing but the ester bond, right? So glycerol and the fatty acids they get combined with each other, bound with each other through the formation of ester bonds by removing 3 water molecules. So these are nothing but the triacyl glycerol. Otherwise, we can call them as the triglycerides. Triglycerides. Otherwise, triacyl glycerol. You should remember that, right? If you see the bond, if it is a monoacyl glycerol, this is the glycerol molecule residue, and this is from the fatty acid, and the box item is the ester group, otherwise ester bond. See here the above picture that shows the triglyceride, that is one glycerol molecule combined with 3 fatty acid chains. So here we will see the bonds. These are the ester bonds. In the below picture we will see only one side chain and the glycerol molecule. It is a monoglyceride. It is the triglyceride, right? And generally we will take fats in the form of phospholipids. So major source of the cell membrane, and the cholesterol, 27 carbon component, which is known as the cholesterol, main component of the all the kinds of fats, otherwise the lipids. And come to the process of digestion of the lipids in various regions of alimentary canal like mouth, stomach and the small intestine. Let us see how the digestion of lipids takes place in the mouth. In general, the mammals' saliva do not contain any kind of lipase enzyme, because the lipase are the enzymes which can break down the lipids into fatty acids. But in the saliva of mammals, there is no lipase enzyme. Okay, so that there is no digestion in the digestion of lipids in the mouth. But in the saliva of the human beings, in the saliva of human beings, there is a enzyme which is known as the lingual lipase. It is known as the lingual lipase, which is produced by the Ebner glands. Ebner glands are the glands which are located near to the tongue, and they secrete lingual lipase. And although the lipase enzyme secreted in the saliva of the human beings, but the digestion of lipids not at all takes place in the mouth, buccal cavity. The lingual lipase which is secreted by the Ebner glands, that is highly active in the acidic pH environment, so that is active in the stomach, that actively function in the stomach, right? Whenever the saliva mixed up with the food, along with the saliva, the lingual lipase also mix up with the food, but there is no proper digestion of the lipids in the mouth. As and when it reaches the stomach, due to the acidic environment, it is highly active and thereby digests the food there in the stomach itself. So that here what we have to remember, no digestion of lipids in the mouth due to lack of lipase enzyme in saliva, but in the human beings there are Ebner glands, they produce lingual lipase, but the enzyme is highly active in the acidic environment, that is in the stomach. These are the few things which we have to remember under the subheading digestion in mouth. And coming to the next portion of the alimentary canal, stomach, digestion of lipids in the stomach, how they are going to take place, right? The initial digestion of the fats takes place in the stomach itself, and here there are two things that facilitate the digestion of lipids in the stomach. Number one, heat condition, otherwise hot environment of the stomach, and the movements which are happening in the integument of the stomach, they are known as the churning movements. Both the hot environment and churning movement make the mixing of the food thoroughly, and thereby it will become very smooth, and smooth food has been formed due to the hot environment and the churning movements of the stomach. And the acidic pH of the stomach also facilitates the digestion of lipids. The pH may be of about 2. The pH may be of about 2. This acidic environment, acidic pH environment that facilitates the lipids to undergo digestion. Right? Now, along with the food which has been entered into the stomach, there will be a mixed up of the lingual lipase within the food. Now, this lingual lipase highly active in the acidic medium and thereby of about 30% of entire lipids is only digested by the lingual lipase, and moreover of about 4 hours of time the food is stored in the stomach. Until that time, how long the food is stored in the stomach, that much time it is highly active and continuously functioning to break down the lipids. For of about 4 hours, 30% of fats has been digested by the lingual lipase. And the parietal cells of the cells of the integument, parietal cells of the stomach integument, they produce gastric juice. In the gastric juice we see gastric lipase. We see gastric lipase enzyme. It also participates, it also plays a major role in the digestion of the lipids. Right? Due to the combined effect of both the lingual lipase and the gastric lipase, all the short chained fatty acids, they may be get digested off. Due to the combined effect of lingual lipase and the gastric lipase, most of the short chain fatty acids get digested. And moreover, one more thing we should remember, that the digested lipids absorbed into the portal vein of the gastric integument, to the portal vein of the gastric integument. Right? So here some sort of lipids get digested off, and the digested lipids absorbed into the portal vein of the gastric integument. And coming to the third portion, which is very, very important for the digestion of the lipids, digestion of lipids in the small intestine. The digestion of lipids almost all completes in the small intestine. The smoothened acid chyme, as and when it re-enters into the initial part of the small intestine, that is the duodenum, there is a release of bile juice from the liver, and then pancreatic juice from the pancreas, bile juice from the liver, pancreatic juice from the pancreas, saccus entericus from the small intestine. All of them they released into the small intestine. As and when the acid chyme enters into the duodenum of the small intestine, there will be a release of bile juice from the liver, pancreatic juice from the pancreas, and the intestinal juice, that is the saccus entericus from the small intestine. All these three kinds of juices they will release into the small intestine, they mix up with the food material. Then what is the use of all these three kinds of digestive juices? Right? In general, in the bile juice, very, very important thing to note and remember is bile juice contain no enzymes. No enzymes are present in the bile juice. Only bile salts are there and bile pigments are there. Bile salts and bile pigments are there. So bile pigments, as we know, the bilirubin and biliverdin, right, which color the fecal matter. Now here bile salts, basically there are glycocolic acid and the taurocholate acid. these two things, glycocolic acid and taurocolic acid, whenever they are mixed up with the sodium and potassium, they may form the sodium potassium salts of the gastric juice, bile juice, right? That is sodium glycocholate, sodium taurocholate, potassium glycocholate and the potassium taurocholate. Okay, although the bile salts are there, although there are no enzymes at all, so that they are unable to digest the lipid material. As the digestive enzymes are absent in the bile juice, this bile juice may not used for the digestion of lipids. But the bile salts, they play a major role in the emulsification of lipids. Emulsification of lipids. What is the emulsification? What is the use of the emulsification? In general, the lipids are always in the colloidal form, large colloidal form. These large colloidal fat substances, lipid substances, they undergo emulsification and modified into small oil droplet like structures. Small oil droplet like structures. And whenever you compare the non-emulsified lipids and the emulsified lipids, non-emulsified lipids not at all undergo a proper digestion with the lipase enzyme. As and when the lipase enzyme act upon the non-emulsified lipids, that may not digest properly the lipids. But the emulsified lipids, they readily undergo the digestion of lipase activity. That is, emulsification is a process where there is a conversion of large colloidal substances into small oil droplet like structures, and thereby this emulsification facilitates the proper digestion of the lipids with the help of the lipase enzyme. That is, they are readily undergo the digestion with the lipase. Why? Because why there is a difficulty in the digestion of the fats with that of the lipase is all the fats are water insoluble. They are hydrophobic in nature. They are hydrophobic in nature. Whereas the lipase is hydrophilic in nature. So that the fats are unable to dissolve in the water, but the lipase is readily dissolved in the water. So that the water soluble lipase unable to digest the fat that much easily. It is very difficulty to digest the hydrophobic natured fats, right? But after the emulsification, the large colloids, they will be degraded into small droplets, oil drop like droplets, and thereby they will be distributed all over the water, all over the surface of the water, and thereby the bile salts and the phospholipids, they are the amphipathic in molecules. That is, they have the hydrophilic and hydrophobic groups. So thereby they will bind on one hand with the oil droplets, another hand with the lipase, and thereby they brought together the enzyme and the substrate, and finally lipase is easily soluble, and so that it can penetrate into the water medium, ambient medium, and surrounds the oil droplets, and finally it easily digests the lipids, right? So that due to the emulsification process, there will be enhancement of surface area of the substrate by the emulsification, right? It facilitates the digestion of the lipids by the lipase enzyme. Okay, so these lipoproteins, otherwise the enzyme, lipase enzyme, colipase enzyme, it also helps in the digestion of the lipids, and this is also amphipathic enzyme which connects the, which plays a major role in the interfacial activation. That is, one side it can bind with the fat, another side with the lipase, and lipase has active sites. These active sites are get opened, exposed by the colipase. So such a phenomena is known as the interfacial activation. Interfacial activation. In the diagram we will see here, this is a large fat molecule, and here this side hydrophobic and this side hydrophilic, and finally this large fat get digested into, modified into small oil droplet like structures which are known as the emulsified droplets, emulsified fats, right. So it is a hydrophobic lipid, and after the attachment of bile salts and the phospholipids, they will become like this. So there is a increased surface area for the action of the lipase enzyme, right. Here we have to remember, one is the emulsification, first thing is the emulsification, right. So how the emulsification facilitates the action of lipase on the lipids. So both the pancreatic juice and the saccharicus, they have the lipase enzyme, but it is highly active in alkaline pH of about 8. It is highly active in the alkaline pH, right. Then if you see how the lipase can digest the lipids. For example, this is the glycerol molecule and this is the glycerol molecule, and these are the different glycerol and 3 fatty acids, let us assume. And the pancreatic lipase breaks the ester bonds of first and third positions. See, this is the first position and this is the third position. Here it breaks. As a result, we will see 2 fatty acid chains will be released and glycerol and 1 fatty acid, monoglyceride is released. And at the same time, phospholipase A2 of pancreatic juice and the phospholipase of the saccharicus, both of them, they act on the phospholipids in the presence of calcium ions, convert them into glycerol, fatty acids, phosphoric acid and the choline. Phospholipase A2 and phospholipase, they both of them act upon the phospholipids in the presence of calcium ions, thereby the end products are, as a result of this, the end products are glycerol, fatty acids, phosphoric acid and the choline. These are the end products. Then if you see the entire process of the digestion of the lipids in the small intestine, here triacyl glycerol or triglycerides, nothing but the lipids, they enter into the small intestine, and here it is a mixing of bile juice, there are bile salts. Bile salts make the emulsification of the lipids, okay. And for the lipase and colipase activity, we require the optimum pH is 8. Thereby the lipase and the colipase, both of them, they act upon the emulsified lipids. Finally they degrade the lipids into fatty acids, glycerol, monoglycerides and the diglycerides. Finally we will get fatty acids, glycerol, monoglycerides and the diglycerides. This is the digestion of the lipids in the small intestine. And thereby, if you see here, there is a large fat molecule with the mix of the bile salts. There will be a formation of oil droplet like structures which are known as the emulsified lipids, and generally these are the triacyl glycerols or triglycerides. Due to the cutting of first bond and third bond, we have free fatty acids and monoglyceride, okay. They are readily digested into the normal small intestine. right? In the final stage, after the complete digestion of the lipids, due to the combined effect of pancreatic lipase, colipase, phospholipase, the emulsified lipids they get digested into fatty acids, glycerol, monoglycerides and the diglycerides. This is all about the digestion of lipids in various parts of the body. Thank you one and all.